So, okay. So Shamika, you are right now the only one. Anything from the previous class that you want to take up, or else we will move ahead with our today's class. Kindly give
And
me a
right now,
confirm.
I'm not having anything to ask.
Okay, okay. I just wanted a confirmation if I'm audible and all. Okay, in that case, we'll just move ahead with today's class. We can then discuss anything at the end of the session as well. Just give me 1 minute. So, as of last class, we finished our discussion with the B cells, and then we moved on to understand that whatever are the cell surface molecules which are decorated onto the T cell membrane. And we also briefly saw the property where it's a property of the T cells that they are self-MHC restricted, right? And there was a Nobel Prize winning experiment that we discussed performed by Doherty and Zinckernagel, wherein they demonstrated this experimentally that it's only if the antigen is presented along with the self-MHC molecule we will have an immune response. And if you're talking in terms of T cells, the cells of interest here were the cytotoxic T cells, and we saw that they were bringing about a killing function. Now, before we move ahead with further discussions onto the different properties and features and the effector responses of the T cells, we will first understand the process of development of your T cells, right? So, we discussed this from our very first class itself, where we saw that the T cells So for the B cells, the development was somewhat simpler because there was no additional step of it getting migrated into another organ for it to become a fully functional, a mature, or a naive B cell. However, for the T cells, the origin is from the same bone marrow, but there is a involvement of the step of migration wherein they need to reach the thymus, and the thymus is the ultimate organ from where we have the naive T cells exiting, and eventually they're immunocompetent. So now by this class, I hope certain terms that I use that you are clear of it, right? So immunocompetent, which means that it can now recognize a particular antigen. Its antigenic specificity has been determined. Okay. So if you look here for again, we also discussed this last time that once we have done with our discussion of B cells, there are many things that you will find overlapping with the development in the process of T cell development. So wherein we will have some say the process of selection that we discussed last time. That is one topic that we will discuss in a lot of detail in today's class. We had recombination events happening to rearrange the antibody genes, right? Present on the surface of the T cell or of the B cells or when they are produced in the soluble form. Similarly, we will have recombination events going on during the process of development of the T cells as well. So if we remember everything from the B cell, the topics relevant to B cells, we will be able to move through this comparatively quicker, and it will also be easier for us to understand everything that is happening in the T cells as well. Starting from the beginning, so every cell in the blood, they start their life as the hematopoietic stem cells. And if you follow from the first classes of this very initial classes of discussion, that there are two progenitor lineages. One is the myeloid progenitor and the lymphoid progenitors. We have all the lymphoid cells or the lymphocytes coming from the lymphoid precursor cells. So this is what has been mentioned here. So inside the bone marrow, we have the hematopoietic stem cells that will eventually differentiate into a common lymphoid progenitor cell or a precursor cell. So from here, we will either be having the B cells or the T cell differentiation, right? Now, there are certain markers which are specific to every cell that we talk about, and these markers are what so it's like an ID card for all the cells. So if a cell has all these three markers, we classify that as hematopoietic stem cells. And as we follow through this developmental process, we see that as the hematopoietic stem cell is going to differentiate into ultimate T helper cell or the cytotoxic T cells, you will witness that a lot of markers are changing. There are some which are going to cease its expression, some are going to find new expression, and eventually, based on all these identities, we designate a cell into different types of it, right? So the first cell that it starts from, every blood cell starts from, is the hematopoietic stem cells, and we have c-KIT, CD44, and CD25 markers present on the surface of this hematopoietic stem cells. So what is c-KIT? c-KIT is basically a receptor. It's a receptor to the stem cell growth factor. So whatever is the growth factor in the near vicinity, we have hematopoietic stem cells expressing c-KIT, which is able to respond to the growth factors in the environment, and it will ensure that this cell keeps on growing further, as the name suggests. C44 is an adhesion molecule. For homing means that it's defines the identity, defines the localization of the molecule. So we have certain pockets, certain defined regions wherein we will be finding a hematopoietic stem cells. And how does a stem cell know that it has to reside in that region? Is because of the presence of some homing adhesion molecules, which is CD44. Apart from that, we also have CD25. CD25, somewhat contrary, is a beta chain of the IL-2 receptor. So IL-2 is we briefly discussed that there are certain interleukins of different types, and IL stands for interleukin. Now, interleukins can be of different types, and they have been numbered I think they have been numbered in the order of their discovery. So we have IL-2, which is a cytokine, interleukin 2. The receptor for it has two chains, the alpha and the beta chain. But in from the hematopoietic stem cell onwards, the beta chain of the IL-2 receptor is present on the HSCs, the hematopoietic stem cells. It then differentiates based on the signals that it receives from the environment, the stromal cell networks in the bone marrow, and differentiates into a common lymphoid precursor cell. The lymphoid precursor cells undergo migration from the bone marrow inside of the thymus by the 8th to 10th week of gestation. These times and these are the times gestation means when you are in your embryonic stages. So from the 8th to ninth gestational week of the mouse. So all the reference points that you might encounter in today's class have mostly been discussed in the book in context of the mouse. So this is the 8th to 9th week of gestation in the mouse, right? By this week, it migrates inside of the thymus and rest of the processes. The most of the So from the this lymphoid precursor cell to formation of the mature T cells, all of this, if you see, is taking place inside of the thymus. So that's why we have also seen that whenever thymus is subjected to certain ablation, maybe if thymectomy is performed, the T cell population is majorly compromised, and we have immunocompromised model organisms that are formed because we do not have any T cell response present in those, because all the essential processes from the very beginning till the formation of mature T cells is entirely happening inside the thymus. So in the thymus, we have the progenitor cells, the lymphoid precursor cells coming in. The cells that now cease to express CD44 but continue to still express c-KIT are designated as the T cell precursors or thymocytes. These cells will further cease to express c-KIT, and only CD25, which is a IL-2 receptor, is going to be expressed. Such cells are called the pro T cells or the double negative cells. So what happens when we discussed about the T cells, we say that we have a TCR, and along with TCR, we have two co-receptor molecules based on which the subpopulation of T cell is defined, right? CD4 and CD8. So ultimately, we have the mature cells which either have CD8 or CD4, right? But there comes a transient period where we have both of these four receptors present on the same cell. So that stage is referred to as the double positive stage. So prior to that, when either of these co-receptors are absent, that cell is called as the double negative cell. Now for a double so for a double negative cell, we understand that it continues to have the IL-2 receptor, the c-KIT and CD44 markers, surface markers are now downregulated. So this cell is known as the pro T cell or a double negative cell. Inside of the pro T cell, now if you look here, until this point, there was no expression of the RAG proteins. So RAG proteins, coming from the lectures related to B cell, RAG proteins were important for the purpose of. So I think by the class 8, I can ask some questions from the previous class. So RAG proteins, we saw that these are very important for the purpose of. Shamika, you're the only one in the class, so I'm expecting a response from you. You have been very regular throughout, so I expect that you might recall from the previous classes. Are you
Yes,
following
ma'am.
with me?
Yes, ma'am. I am following with you, ma'am. I think that I'm not very much sure, but I think that RAG is the one which recognizes some expression sequences, and then it
Hmm?
is helping in making the cuts along something. If I'm not wrong.
No, you're not wrong. I think it's just that you are not able to explain it properly. Okay, you are right. What you're saying. So expression sequences, we call them signal sequences, right? The RSS sequences. So RSS sequences were getting recognized by two lymphocyte-specific enzymes, RAG1 and RAG2. And we also discussed that RAG can be a marker for your lymphocyte, because if among all the cells of the body, RAG expression is only present in your lymphocytes. You've been right. It's just certain terminologies here and there, but I hope you will be able to follow through that, right? So if you remember from B cells, when the B cell genes had to undergo recombination events where the VDJ recombination was happening, we had certain signal sequences and certain enzyme cutting at those signal sequences so that they can be eventually joined together in different combinations, right? So RAG was the enzyme that was performing that function, and we discussed in the last class that in a very similar mechanism, we have VDJ recombination and VJ recombination also happening inside the T cells, right? And that is how they are also generating the diversity. So, the first to rearrange, very similar to what we saw for the heavy chain of antibody, we saw that the heavy chain rearranges first, and the heavy chain is having DJ joining first, and then the V segment joins to the DJ combination. Similarly, for T cell receptors, we discussed this that it is the beta chain that undergoes VDJ joining and the alpha chain undergoes VJ joining, right? So, what the DJ joining chain always rearranges first. So, in your B cells, we had the heavy chain rearrangement happening first. Now, for your T cells, we had the DJ joining, which is happening for your beta chain happening first. So in the pro T cell, which is a double negative cell because it has no CD8, no CD4, just the IL-2 receptor is getting expressed. We have the RAG expression that in these enzymes responsible for recombination, they are switched on at the stage of pro T cell. This cell first undergoes DJ joining. Once it has undergone DJ joining, it becomes a pre-T cell, right? Now, what is present in the pre-T cell? c-Kit and CD4 cease to express here. So we still have the, so let's just, we'll write that it continues to express the CD25 molecule, which is equivalent to the IL-2 receptor. So now the pre-T cell also undergoes the joining of V region of the beta chain. So V beta joins to DJ segment, which means that now this cell, this pre-T cell, has a fully rearranged beta chain of your TCR. Now beta chain is in place, and if you remember again from the B cell discussion, we had a surrogate light chain joining with your heavy chain that has been arranged. Similarly, in the T cell, we have with the fully recombined beta chain of TCR, we have a molecule joining in called the pre-T alpha. Pre-T alpha, this is so this is like an alternative for the alpha chain that eventually is going to undergo recombination separately, and eventually on the mature B cell we have the alpha chain that the recombined alpha chain joining with your recombined beta chain. It's the alpha and the beta chain that together forms the TCR. But till the time we do not have the alpha chain in proper recombination in place, what joins with this fully recombined beta chain is a temporary alpha chain which is called as the pre-T alpha. So, so in your pre-T cell, so from the pro-T, what has changed? We have a fully recombined beta chain with which we have a pre-T alpha joined. Along with that, we have its association with the CD3 molecule. Again, from the previous class, we saw that CD3 is a signaling molecule which is associated with your T cell receptors. So this is a very important junction in the development of T cells. Why so? So from here, let's just first understand this point, right? So this cell here, this pre-T cell here, along with the CD3 and pre-T alpha and beta, this is the cell surface receptors present on this pre-T cell. So we have the pre-T alpha, which has formed an association with your TCR beta, and these are the CD3 molecules, if you remember from the last class. This association gives very important signals that has multiple roles to play. The first signal that it does is it stimulates proliferation, which means that this particular combination of TCR beta, so as you would remember, multiple V segments can join with either of the multiple D segments that arrange it, that is present, and similarly is for the J regions, right? But whatever is the specificity that has been formed after the VDJ joining, the same cell is going to undergo proliferation without getting into recombination again, right? Which means that the same TCR is going to form, TCR beta is going to form again and again as the cell proliferates. And why is it now undergoing proliferation? Because in the pre-T cell, because of the association of the pre-TCR with the CD3, this is a signaling event that happens. It undergoes proliferation. The second thing that comes up is that, so you have the so this signal also tells the pre-T cell that I now have a fully rearranged, a functional TCR beta chain. So the beta chain do not have to enter subsequent rounds of recombination. Do I have people joining? Right? So one thing, the cell enters into proliferation once again, and also it will not undergo further recombination events for the beta chain. This also stimulates the expression of CD4 and CD8 co-receptors, and along with that, it sets a permission for your TCR alpha chain to now get a chance to undergo recombination. So we have two things. So here we discuss. So this is the first stimulation. And this is also, so at the same time, one cell is getting an instruction that I do not have to rearrange the TCR beta any further, which also forms the basis of your allelic exclusion, right? This we have discussed in B cells. So in a very similar manner, allelic exclusion is also happening here. Here we are talking about the case of beta chain, right? The other signal that comes up is, so once we have this proliferation happening, as we have more cells coming in picture and they are getting more signal to proliferate further. So this one cell divides to the next. This second cell also further is stimulated to undergo proliferation. Now all of this with the same kind of beta chain on its surface, right? Alongside what is happening, that after subsequent rounds of recombination, all of these cells also have the RAG expression going on, because if you look here, RAG expression is already in place, but it is only the beta chain that is still undergoing recombination. Alpha chain requires a permission to start undergoing recombination. So this signal allows your alpha chain recombination to begin. But prior to the permission given to alpha chain to begin, this one pre-TCR has already divided into multiple cells of its same kind. What is happening by this process? Now we have the same cell, pre-T cell, in, say, if it started as one, now we have 10 such cells before the permission of alpha chain recombination could begin. We will see how this permission is given exactly. But before the permission is given out, we already have 10 cells with the same type of beta chain on it. Now, all of these cells independently is going to get a chance to rearrange its alpha chain. So here comes again uh, the phenomena of probability takes its chance. So the chances of all your beta chains to assemble with exactly the same kind of rearranged alpha chain is very less, because recombination is a random event, and it is most likely that all the 10 pre-T cells are going to undergo different recombination event of the alpha chain, and eventually the alpha chain that associates with the pre TCR's beta chain, they are all going to be different, and hence all the 10 cells will now have different specificity for different antigens. Is this part clear? Because it's a lot of simultaneous events that is happening. So first thing that happened is that the pre-TCR assembly with CD3 happens. This cell is known as the pre-T cell. Pre-T cell has no co-receptors on its surface. The assembly, the signaling by the pre-TCR and the CD3, it signals that this cell now needs to undergo proliferation. And it also signals that the alpha chain can begin recombining. But prior to the process of alpha chain recombination completing, we have the pre-T cell already divided into multiple cells of the same kind, multiple cells having the same beta chain of the TCR. Now, all of these cells, when they independently undergo alpha chain recombination events, different alpha chains are going to combine with the same kind of beta chain of your pre-T cell, and eventually the next cell that forms, the pro-T cell, here you see that the alpha has come into picture. This is going to, so pro-T cells, which has started from one type of pre-T cell, has different alpha chains joined to the same kind of pre-T cell, right? And here the cell also received the signal for expression of CD4 and CD8 co-receptors. So the pro-T cell expresses alpha chain, and also with that it now expresses both CD4 as well as CD8. So that's why this cell is referred to as a double positive cell. Right? Now, coming slightly back to how the permission is given. So what happens in the pre-T cell that has rearranged the beta cell, we have recognize some intrathymic ligands here. So this signaling comes into play because a pre-T cell can recognize some of the ligand molecules being expressed by the cells of the thymic stromal network, right? So a lot of epithelial cells, nerve cells, dendritic cells, a lot of other cells are present inside the thymus. And what is this signal coming from? This signal is coming from the association of this complete receptor along with the ligands being expressed by the thymic epithelial cells. Apart from that, what happens that because of this signal, the RAG product is degraded, right? So we do have the RAG gene expressions present, but the RAG2 product, the enzyme RAG2, it quickly gets degraded as soon as it forms in your pre-T cell. Now, as the pre-T cell undergoes the subsequent rounds of division, gradually we have the accumulation of RAG2 product. And when a threshold value of RAG2 is reached, only then your alpha chain enters the rounds for rearrangement, right? So alpha chain rearrange only as RAG2 reaches a certain threshold level. And that's how it is ensured that the alpha recombination event only happens after some rounds of cell proliferation has already occurred. Now this double positive cell, it has to undergo a process of selection, and eventually this differentiates into CD8 or CD4 plus mature but naive T cells. These cells get inside the blood from where it can get into different peripheral tissues. Apart from that, one more thing. So we have discussed here for alpha beta type of T cell, which is the most dominant population of T cell in the circulation. But apart from that, as we have also discussed, that there is another type of TCR, the del gamma TCR, right? So where are they coming from? So they also until this point they follow the same path, right? So the pro T cell here, instead of productively rearranging its beta chains, if both the alleles of beta chain fail to rearrange properly, we then have its it's del chain, del gene that gets a chance to enter the recombination event. If the del gene enters recombination, what associates further instead of alpha is the gamma chain, and we saw that it's the alpha and the gamma genes are are arranged in a manner in the same chromosome, that if alpha has formed, the gamma gene has been spliced off. But over here, a alternate possibility is that if the beta chains fail to rearrange properly, we have the del gene that gets a chance to rearrange, and such pre-T cell where the del gene has undergone recombination and is expressed on the T cell, we have the gamma gene now that associates with this particular uh, uh, T cell, and the ultimate make of this T cell is a TCR of the gamma del type. Right? Okay. So anything so far that you would want me to go over again, or else we'll move ahead? We will next get into our discussion related to the process of thymic selection. So a very brief overview that what happens. So this thymic selection, of course, it is taking inside the thymus, and the thymic stromal cells, they play a very important role. Two kinds of selection happens. We saw that the MHCs essentially have to be self-MHC, the T cells have to be self-MHC restricted. So that is coming into play because of the positive thymic selection that happens for the T cells that can only recognize antigens that are being presented by self-MHC molecules. So only the T cell that are going to get a chance to enter into the circulation are the T cells that recognize not antigens just bound by any MHC molecule, only the antigens that are bound by the self-MHC molecules. If this complex is recognized by a T cell, this T cell is going to get positively selected and get a chance to further become a single positive CD8 or CD4 plus cell. And at the same time, after the positive selection has occurred, we have the same T cells subjected for negative selection. So, in negative selection, something is going to get eliminated, right? So, this eliminates T cell that reacts to the self MHC molecules or the self MHC conjugated with your self antigen complex. So, what happens here? So, your MHC molecules are also they are also immunoglobulins. So they on on its own, these molecules can be recognized as an antigen. So if we have any of the positively selected T cell that can recognize your self MHC molecule itself as an antigen, those cells are eliminated. And similarly, if we have any of the self antigen being presented alongside your self MHC molecule, that now also gets deleted or negatively selected. Such cells will not reach inside your circulation.
So, mom, the cell first undergo positive selection and then undergo negative
Yes.
selection, or only one will be happening?
No, no, both the things happen. First, they are selected by positive selection, where the self MHC restriction is achieved. And next, we have the self tolerance, which is achieved by the process of negative selection. So, positive first, and then it is subjected to negative selections.
Okay, ma'am. And ma'am, in drag expression, when it is switched on, so firstly, in the beta chain rearrangement, in that case, rag 1 is responsible, and in the second time, while for alpha chain, the rag 2 is responsible. Is it
No,
so? Or
no,
rag
no,
2
no.
is
So if you remember from the mechanism of VDJ recombination, RAG1 and RAG2, both the enzymes together are bringing about the different gene segments uh, together so that the intervening sequence can be spliced out, right? So both the genes, both the enzymes, RAG1 and RAG2, have a equally significant role while any recombination event has to occur. It is by mechanisms. So, in the pre T cell, which already has the beta chain rearrangement, which is now completed, the expression of RAG genes continue to occur. By some other mechanism, the product of RAG2 is quickly degraded. So, the RAG2 enzyme is quickly degraded inside your pre T cell. So, say, अगर रैग एक्सप्रेशन नॉर्मल होता, अगर डिग्रेडेशन नहीं होता, तो 10 रैग 2 मॉलिक्यूल्स बनते, and 10 rag molecules are say required to bring about effective recombination. But because now RAG2 is getting quickly degraded, of the 10 molecules formed per second, only 2 molecules remain per second. These are just random figures I am giving you, right? So 2 molecules remain and rest of the 8 are getting degraded. So this 2 alone is not effective to bring about recombination while the proliferation signal is still on. So recombination अब alpha का ही होना था क्योंकि beta is already done. The next that should follow is the alpha chain rearrangement, but RAG2 is not there because its products are getting degraded. ना धीरे 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 ये रैक टू बनता गया, डिग्रेड होता गया, फिर बना, फिर डिग्रेड हुआ, फिर बना, फिर डिग्रेड हुआ, बट इवेंचुअली when the threshold of 10 molecules of RAG2 is reached, by the time that is reached, this pre-T cell has already undergone multiple rounds of proliferation. इस सेल से मल्टीपल डॉटर सेल्स बन चुके, अब जाके रैग टू एक्सप्रेशन 10 तक पहुंचा। तो अब जब ये 10 पहुंचा, अब फटाफट से अल्फा चेन का रिकॉम्बिनेशन हो सकता है, और फिर अल्फा चेन रिकॉम्बिनेशन होने के बाद जैसे ही ये चेन बीटा चेन के साथ एसोसिएट हो गया, अब प्रोलिफरेशन स्टॉप हो जाएगा। फिर प्रोलिफरेशन इनहिबिट करने के सिग्नल्स काम पे आ जाते हैं, राइट?
मैम, सो मैम रैग 2 यहाँ पर प्रोडक्ट नहीं है, इट इज एंजाइम ना?
हम्म RAG is also the name of the gene. The enzyme that forms from this RAG gene is also called RAG. So it's just we just designate it as gene if we are writing it in small letters, and if we are writing something in capital letters, that means we are referring to the gene product. तो अगर यहाँ पे, because यहाँ पे आपका रैग कैपिटल्स में लिखा है, so they are talking in terms of the gene product that is being formed, right? So that's just a nomenclature of how we usually refer to things in science. But rag is the name of the gene, it is also the name of the enzyme that's being formed.
Okay, ma'am. And ma'am, T cell, prior to pro T cell, it is written V beta, D beta, and J beta. So it is present in the earlier phase of the pro T cell. And when it got matured or got double positive, then it is having V alpha and J beta, right?
See, I also did not understand that why they have written it J beta, because it has to be J alpha. Even I found that this thing, but it has to be J alpha. That is what the text of the book also specifies. But I will also look into it. So, but as far as the understanding part is clear, this has to be J alpha, right? So we have the B and J of the beta chains joining first in your pro T cell. then the V segment is going to further join alongside this in the pre-T cell. And if this pre-T cell now has to become a pro-T cell, alongside expression of the co-receptors, both the co-receptors, the alpha chain recombination has also occurred. So this is the overall make of a TCR, because now it also has the alpha chain and it also has the beta chain. So this has to be J alpha. That is what the text also mentions. I don't know if it's a say that the book has a misprint is a big statement, but as far as I could understand from the text, this is what the authors mean.
Okay, ma'am. And ma'am, in the below diagram of it,
Mm-hmm.
which part is responsible for signaling of all these four?
See, CD, this thing that you see, so
Ma'am,
this CD,
pre-TCR. Yes, ma'am, this.
sorry, over here. Mm-hmm.
No, ma'am, below diagram only.
Which one? This one? This one?
No, ma'am, the below that you have inserted.
Here.
Yes, ma'am, this one.
Yeah, yeah. Tell me.
Ma'am, the joining of which two chains is responsible for inducing the signals which are performing the following function?
It's not any of the chains that are joining amongst each other that is leading to this signal transduction, right? So this is a pre-TCR. So a TCR, if you remember, is in itself, along with TCR, alone TCR cannot give any signal, right? So CD3, so these chains are all of CD3 and they are present in association with the TCR molecule. Now, there might be certain cells in the thymic stromal network which are presenting some antigens on its surface. So, for recognition of the antigens on the surface of certain stromal cells in thymus, your pre-TCR alone cannot mediate any signal. We need association of your pre-TCR along with the signaling molecule, the CD3 molecule. So whenever we have a ligand binding to this pre-TCR, which is in association to the CD3 molecule, we have the signals that can guide the following processes. So it is not your pre-TCR joining with the CD3 that is initiating this signal. Some sort of interaction, as I saw, this is guided by the intrathymic ligands.
Okay, ma'am, got that point.
Just a second. Give me 1 minute, right? Yeah. So, continuing with our discussion of thymic selection. So, as we were saying, that selection happens to ensure self-MSC and restriction as well as self-tolerance. Now, there was a small experiment again performed by Zinker-Nagel, which demonstrated this phenomena of self-MSC restriction. What happened? So, if you remember, so this experiment that we discussed, it took certain strains of mice, right? So, again, over here, what he did, he crossed two different strains, say strain A and strain B of mouse, and the progeny of it eventually is going to have both these molecules getting constituted in its MHC. So the progeny, so it's simple. So it's A and B coming together in the progeny as AB, right? So this ABF1 is of this particular strain now. AB, both of these things are present. This mice was experimentally undergone thymus removal. So this process, thymectomy, is nothing. It was the surgical removal of the thymus. So thymus removed means all the T cells, all the thymic stromal cells, everything is removed. And whatever are the cells in the circulation or everything, those have also been removed by lethal X-ray irradiation. So eventually, this mouse that we have, this is the same progeny, but here from the B parent, they took thymus outside the B parent and they grafted the thymus inside the same mouse which underwent thymectomy and lethal X-ray irradiation. So thymus आ गया. अब thymus ही तो सिर्फ नहीं हटाया, जो भी इसके अंदर सेल्स थे वो सब भी हटा दिए गए थे. तो यहाँ से जो भी सेल्स रिकवर किए इन्होंने, उन सेल्स को वापस डाल दिया और थाइमस डाला सिर्फ बी वाले पेरेंट का. So whatever are the hematopoietic stem cells, they are still the same as the original mice that underwent these experimental processes. It's just the thymus that has now come from strain B. Thymus के अंदर दो चीजें हैं. One, the stromal cells and the developing T cells. T cells इन्होंने सेम डाल दिया, थाइमस डाला इन्होंने सिर्फ बी पेरेंट का. मतलब क्या कि सिर्फ जो स्ट्रोमल सेल्स थे वो पेरेंट क्या है वो उन्होंने इस ऑर्गेनिज्म को ऑलरेडी हटा दिया था. नाउ दे इन्फेक्टेड दिस माइस विथ अ वायरस. आफ्टर वायरल इन्फेक्शन, they now dissect out the spleen of the mouse and they isolate the cells from the mice, the spleen of this mice. So inside the spleen, it's a lymphoid organ. So we will have all the effector T cells. So amongst the effector T cells, we will also have the cytotoxic T lymphocytes. These cytotoxic T lymphocytes were now incubated in a cell culture where so we either have cells of strain A infected by this particular this same virus infecting strain A cells or the same virus infecting strain B cells. Now, when the CTLs isolated from this mouse were incubated with either of these two cell cultures, and it was noted that the killing only happened for LCM infected strain B cells. It did not happen here. Now, if I do not explain this, do you understand why this happened? You can follow through whatever is written on the image. And is it clear that why here we had the killing and here we did not have a killing? Considering the fact that we have discussed what is self-MHC restriction. You can take 2 minutes and just tell me if you can explain, and even if you cannot explain, I'll explain it to you. So what happens here is that
Just give me 2 minutes. In the second stage, where the strain B thymus has been injected, has been grafted to the mouse, and it is having its own hematopoietic stem cells, right? Of H2A and B, having both the strains, right?
The
So if
thymus that has come in has so because it is coming from the parent B. So parent B का जो thymus होगा उसमें hematopoietic cells भी सिर्फ B वाले होंगे
यस
और
मैम, वही
हम्म
होंगे, तो इसीलिए जो है दैट्स व्हाई इट इज ओनली एबल टू किल दी LCM इन्फेक्टेड स्ट्रेन बी सेल्स एंड नॉट ऑफ ए सेल्स, बिकॉज ए सेल्स के उसमें प्रेजेंट नहीं होंगे, ऑल्दो उसके पास में हेमाटोपोइटिक स्टेम सेल्स और बोथ उस ऑफ स्ट्रेन ए एंड बी प्रेजेंट हैं।
तो ए एंड बी तो इसमें प्रेजेंट है क्योंकि हमने ये डाला है hematopoietic stem cells which is a make of both A and B. We have put that in. We have only put thymus of the strain B.
सो मैम वहां पर MHC रिस्ट्रिक्शन भी जो है वो उसी के
Right.
सेल्स से रिलेटेड ही हो रहा होगा ना, बी मतलब बी रिसेप्टर्स, बी स्ट्रेन के जो भी सेल्स के रिसेप्टर्स होंगे तो उससे रिलेटेड ही जो है उसका MHC मॉलिक्यूल का रिस्ट्रिक्शन भी हो रहा होगा, मतलब वो उन्हीं के कांटेक्ट
Exactly.
में आ रहा है, सो दैट्स व्हाई वहां पर स्ट्रेन बी से ही वो यूज टू हो रहा है, ए से नहीं हो रहा है, दैट्स व्हाई इट इज नॉट एबल टू किल
Exactly.
ऑफ स्ट्रेन ए से।
Absolutely right. Absolutely right. Right. So you understood this fact, right? कि यहाँ पे क्योंकि thymus has the stromal cells that is educating your developing T cells about the self MHC molecule. Now, if the thymus is coming from strain B, whatever is the MHC type of the stromal cells of this thymus is of the B type. So, MHC of strain B. So, whenever we have any recognition of antigen that is happening, this mouse now only considers, because here the selection has only occurred for a T cell that can recognize peptides conjugated with MHCs of strain B. So now if we subject it, we isolate these T cells and we subject it to LCM infected A and B cells. So strain A B cells will have strain A MHC and strain B cells will be having strain B MHC. The CTLs that we could isolate, because the thymus was of strain B, these are restricted to MHC of strain B. Since they are restricted to MHC of strain B, they could only bring about killing when we have LCM infected strain B cells. So this is how self-MHC restriction was experimentally demonstrated, and it was also shown because if we have also put hematopoietic stem cells of A and B both types, right? So where is this education happening? That part is also clear because inside the thymus, if the whatever is the stromal cell MHCs of the thymus, that is what these cells are going to learn as their self MHCs, right? And similarly, Mhm.
So, ma'am, the cells here are only having CD8 receptors on their surface, or also they are having CD4 plus.
See, the experiment that has happened, they are talking in terms of the CD8 positive because those are the cytotoxic T lymphocytes. But if something similar could have been done for T helper cells, because T helper cells के केस में आप directly evaluate नहीं कर सकते. क्यों? क्योंकि T helper cells के effector response में हमें cytokines मिलते हैं. यहाँ we can evaluate killing. But over there, which cytokine, and there are many cytokines, right? We cannot also say that whenever a T helper cell is going to get activated, a particular, say, this X cytokine is always going to be produced. So we can just quantify them. That is also not the case because different antigens will bring about activation and release of different cytokines. So how will you quantify that? Experimentally, to do that, it is a slightly more complicated and the results that come out will not be very conclusive. So we can but quantify the killing phenomena. So the experiment is done for your CD8 positive cells, but we will just come to it. It has also been shown by some other mechanism that MHC is important for your development of CD4 as well as CD8 positive. Just hold on. We'll just come to that. So anyways, experiment is for your CD8 positive cells, but the similar phenomena also applies to CD4 positive cells. Now over here, this served as a control. If they would simply take the, instead of thymectomy and X-ray radiation, if simply they would have isolated cells from the spleen of this progeny itself. Now the thymic stromal cells will also be having MHC of A and B type. So when, if they would have done this for this particular progeny in itself without thymectomy and all of these things, the MHC, the T cells can recognize MHC specific to both these strands because ultimately it's the progeny individual, right? So this group served as the control for the above experiment. Now, so we understood that how people have looked into the that some phenomena like that occurs. Now, to reason it, the mechanism for selection, how that is happening. So this T cell precursor is there. They undergo rearrangement of TCR genes, and eventually we have an immature thymocyte, which is CD8 as well as CD4 positive. So this is a double positive cell. Now this double positive cell first undergoes positive selection for cells whose receptors binds MHC molecule. So class 1 or class 2 MHC molecule that is presented to the double positive cells by the thymic epithelial cells, this, this recognition is going to give a protective signal. So we have a double positive cell here and we have MHC expression by the thymic epithelial cells. So all nucleated cells of the body obviously has a class 1 MHC molecule expression, and thymic epithelial cell being an antigen presenting cells also expresses class 2 MHC molecule. Now if the double positive cell can bind to either class 1 or class 2 MHC molecule, this binding that you see here is going to signal a protective information to these cells, and they will not be triggered for apoptosis. The cells here that fail to bind either of these MHC molecules, they are all going to be taken for death by apoptosis because they do not interact with the MHC molecules. Now, once, so this over here is giving me self-MHC restriction because anything that is not recognizing the self-MHC molecules are already deleted, right? This is now subjected to negative selection. So the double positive cell, if they have to now become single monopositive, so either CD4 or CD8 positive, now, how does this double positive cell gets into either CD4 or CD8 positive? So if the interaction is happening with a class 1 MHC molecule, this cell becomes a CD4 positive cell. And if the interaction with MHC was of a class 1 MHC molecule, they become the CD8 positive cell. Right? So this is how your self-restricted T cells differentiate into either of these two lineages, that based on which MHC molecule type they could interact with. Now, if we have any of these positively selected cells also reacting or recognizing your MHC in itself, if it is getting recognized, so if you uh, so they have not really drawn it very distinctively, but if it, just imagine that this T cell, instead of recognizing the antigen, is recognizing your MHC molecule itself as an antigen in that case, and if the antigen being presented to it is your self antigen, so in both of these cases these cells are negatively selected and will be triggered for death because they have high affinity receptors for self MHC molecules or self MHC with self antigen molecule. Now, how people have understood that this is this differentiation of a double positive cell into CD4 or CD8 positive cell is MHC guided. If they have certain mice where they have specifically depleted the class 1 MHC molecule, they would see that double positive cells are formed, but only CD4 cells are getting formed. There is no CD8 positive cells. Similarly, if people experimentally specifically deplete the class 2 MHC molecule, They see a double positive cell and a CD8 positive cell, but no CD4 positive cell. So this is how we understand that the CD4 positive cells are also dependent on MHC molecule, and as I was trying to give you here, and in fact, the differentiation of double positive cell to CD4 and or CD8 is mediated by which interacting MHC group was found by the double positive cells, right? So this now how is this happening? So there is always a how to every new discovery that happens in immunology. So people have also tried to reason that how is this MHC binding? That if MHC1 is bound, a CD8 is going to form, or if MHC2 is bound, a CD4 is going to form. People have also tried to reason it using two models. One is your instructive model, where the binding of a double positive cell by a MHC1 molecule is going to give a signal which is called your CD8 engagement signal. Why so? Because binding of MHC1 is only favored by the CD8 co-receptor. So whichever co-receptor is engaged, this engagement signal is eventually going to first decrease the expression of CD4, so CD4 low, and increase the expression of CD8, so CD8 high. And eventually, further rounds of differentiation, the cells become CD negative and 8 positive T cell, or a cytotoxic T lymphocyte, or a cytotoxic T cell. On the contrary, if this double positive cell could engage its CD4 co-receptor to interact with the MHC2 molecule, this engagement is going to eventually further augment the expression of CD4 and decrease the expression of CD8, so that we have a CD4 positive and 8 negative T cell, or a T helper cell. So this hypothesis is termed as the instructive model. And on the other side, we also have another proposed hypothesis to explain this, is the stochastic model, where there is a random depletion of CD4 or CD8, right? So a double positive cell first undergoes a random event where either CD4 becomes low, or and if CD4 becomes low, 8 becomes high, or randomly we have CD8 becoming low and the CD4 becoming high. So because now the expression of just consider here, if now the expression of CD8 is higher, the chances of this particular double positive cell to bind a MHC1 molecule is increased. And since CD4 is randomly down regulated, it is not going to be able to bind a MHC1 molecule, because with CD4 you can only bind MHC2 molecule, and MHC1 binding is not happening. So this cell is eventually taken for apoptosis. Similarly here, if we have CD4 upregulated, binding happens to MHC class 2 expressing antigen presenting cells preferably, and the cell differentiates into CD4 8 negative T helper cells. And if this cell, of course, is now not getting a chance to bind to class 2 MHC molecule, and they will be taken for apoptosis.
The second model is the hypothetical one.
Both of them is under consideration, and we are yet to reach a conclusion that which is true.
Okay, ma'am. And ma'am, in the first instructive model, only
Mm-hmm.
CD4 plus is getting in contact with the antigen, right? And in the second case, the CD4 is getting contacted. Uh, मैम फर्स्ट उसमें जो है
See, antigen contact. So, antigen contact is being made by the TCR. The TCR in both the cases are able to interact with the antigen. If the engagement is of the CD8 co-receptor, if you look here in green, if we have the green co-receptor engaging, we have CD8 engagement signal, and eventually CD8 is going to be higher and CD4 will be low. Ultimately, CD4 will be minus and CD8 positive cells will form. Here, the engagement, so as I'm saying, they should have made it a little more visually easier to understand. So let's just say that this is the interaction happening here. This is a positive interaction. And for over here, it is your CD4 molecule that gets a chance to interact. And this is your CD4 engagement signal. Sorry, I have made a मैंने गलती से same receptor. Okay. If this is the interaction happening, then we have what is called as CD4 engagement signal. And that's how CD4 is first getting higher, 8 is getting lower, and eventually a single positive cell is formed. Interaction of antigen by the TCR is happened as a first step. Now so double positive cells को single positive बनने के लिए either of the four receptors are going to get a chance of interacting with MHC2 or MHC1 molecule. Based on that, they are going to become a monopositive cell.
Okay, ma'am. So, ma'am, then after coming in contact with the MHC, they are proliferating themselves, right? To increase the concentration of CD4 positive cells and either CD8 positive T
No,
cells.
it's the same cell. Because if proliferation is happening till the stage prior to it, right? हम लोग proliferation happens till this phase of the same cell. Once we have the cell that is undergoing thymic selection, it is not undergoing proliferation. During the process of selection, the same cell is undergoing differentiation. So, as a process of differentiation, your expression of certain membrane receptor molecules can either increase or decrease based on the genes that are getting upregulated or downregulated. So, any sort of signal, it is eventually, as I'm saying, so this engagement is happening extracellularly. But we will have some domains here. So if you remember, we have certain intracytoplasmic domains. This engagement here is going to be getting inside the nucleus, and eventually the expression of CD4 plus gene is going to increase. So that is how this is becoming higher, and at the same time, the expression of the transcription factors that are releasing, they are going to inhibit the transcription of CD8 molecule, and that's how CD8 on the surface of this particular cell is eventually going to get decreased. So all of this is happening during the process of differentiation. Okay.
Here we can't say that silencing is taking place.
See how the transcription is going down or how the expression is going up. That is something altogetherly different. Whether it is by gene silencing or it could be by condensing of the chromosome or maybe the transcription factors are going down. That I don't know. We have to look into it. But it was not discussed also. So we can look into it that how exactly this is happening. But by some of the mechanisms that we read in molecular biology, it must be coming up. Can we go ahead?
Yes, ma'am, we can go ahead.
Okay. So once we have understood, so this completes our discussion regarding the development of T cells and how the selection and everything is happening for your T cells. The next that we have to understand is the process of activation of T cells, right? So we have a fully mature but naive T cell. So naive means it has not encountered an antigen. Once it has encountered an antigen, it is now being called as a activated T cell. Now, how is this activation happening? That is what our following discussion will try and understand. So, a very important cell, if you look in the entire adaptive immune response, is your T helper cells, right? Because it is your T helper cell that strengthens, gives signals to increase or decrease both your cell mediated as well as your humoral immunity. So, in cell mediated immunity, I am talking about not just the T cells, but also your cells from the innate immune system. So, if you remember the macrophages, any of the cells of your innate system, all of them were also the components of your cell-mediated immunity, and your T helper cells signals both of them. So your T cells as well as your cells of the macrophages, for an example. So they release cytokines. These are the main players that release signaling molecules, which instructs your cytotoxic T lymphocytes to become cytotoxic to mediate cell killing. And they're also going to release certain cytokines, maybe to instruct your macrophages to increase the phagocytic clearance of your pathogens. At the same time, if you remember in class switching of antibody, we said that the switching depends on the cytokines or the release of antibodies, the splicing and everything that we discussed. All of that was under the governance of cytokines that are derived from your T helper cells. So the cytokines from T helper cells also activate the B cells to produce antibodies. So these are say at the center of your this entire orchestrated adaptive immune system, right? So they are very, very important because they in a way regulate your entire immune response, right? Okay. So for your activation of T cells, so we just discussed the entire story for T helper cell, but the similar things also apply to your cytotoxic T lymphocytes or TC cells. So all your T cells would essentially require two signals and another signal that can be specific for your CTLs and your T helper cells. So the first, let's just finish our discussion around T helper cells and then we will draw a comparison for your CTLs. So the three signals that are mainly required for a T helper cell to get into clonal expansion and proliferation, the first thing is your TCR and CD3 complex interacting with a processed antigen, which is associated with your class 2 MHC molecule presented on your antigen presenting cells. So TCR CD3 of your T cell membrane and processed antigen with class 2 MHC from your antigen presenting cell. These two interactions gives the first signal for your T helper cells to eventually get into expansion. The second signal is from CD28 molecule, again present on the T cell membrane, and the B7 molecule, which is a co-stimulatory molecule expressed by the antigen presenting cells. So B7 is also expressed on APCs. And if you look here in this interaction, there is no antigen that is involved. So that's why this signal is also known as the antigen non-specific co-stimulation. The third signal and the most important signal, so it's nothing like most important because in exclusivity of either of these signals, the cell fails to get into clonal expansion, right? So cytokine receptor, IL-2 receptor that we have been seeing on the T cells throughout the developmental process, IL-2 receptor on T cell membrane needs to get a chance of binding to your cytokine IL-2, which is present in your soluble medium. So any T cell that do not have a properly functional IL-2 receptor also fails to get into further differentiation and proliferation because the binding of IL-2 to the IL-2 receptor is an important signal that mediates your clonal expansion and proliferation for your T helper cells. So some facts related to it. So your dendritic cells are one of the most important antigen presenting cells as they constitutively express your class 2 MHC molecule as well as your B7 molecules. And your macrophages and B cells, which are also expressing class 2 MHC molecule, do not by itself express say enough molecules of class 2 MHC, and they do not at all express the B7 co-stimulatory signal on its surface. If there is an antigen that gets presented on the limited number of class 2 MHC molecule presented on the surface of macrophages and B cell, and this antigen MHC is also getting a chance to get recognized by the T helper cells. So the TCR CD3 complex, if this comes and binds to this antigen being presented with your MHC2 molecules here, eventually that interaction is going to stimulate the macrophages and B cells to increase the number of class 2 MHC molecules and also increase the number of B7 molecules. So if that occurs because B7 interaction with the CD28 of T cell membrane is an important signal. So B7 and class 2 MHC molecule expression levels are increased in presence of an interaction of this interaction. But contrarily, dendritic cells, they always have a high expression of class 2 MHC molecule and the B7 molecules. Another fact remains that this B7 can either be binding to CD28, which gives a positive co-stimulation, or it can also bind to the CTLA4 molecule, and that has an antagonistic outcome. So what happens if the binding of B7 is with CTLA4? This binding is going to give a inhibitory stimulus. So where is the CTLA4 present? It is also present on the T cell membranes, but in much lower number. Now, initially in the resting state of T cell, these are non-detectable. They are in very, very low number. And once your TCR is engaged, so this TCR engagement with this, so this signal over here, this engagement eventually leads to CTLA4 induction. So as soon as we have this signal coming up, CD28 was already there, but eventually the number of CTLA4 is going to increase and the expression is on the maximum in 2 to 3 days time, which means, so this is happening in the first step of activation of T cell. So the activation signal of T cell is also inducing an expression of CTLA4, and its expression is maximum by second or third day of T cell activation. Now, can you have a guess at why that is happening? Because this is an inhibitory molecule, and we discussed that the T cell responses are very short lived to ensure minimal damage caused by a reactive immune system to the own body. So CTLA4 in this way is serving as a mechanism of minimizing or eventually decreasing your T cell response, that जैसे जैसे time बढ़ रहा है, you have the effector molecules coming out in place, and now the body has almost been able to tackle the challenge. So CTLA4, which was all the while they were getting increasing in number, now they can be bound by B7 in a competitive manner. Competitive manner, that if B7 is binding to CTLA4, it is not getting a chance to bind to CD28. So eventually this co-stimulation is lost. And as this co-stimulation is lost, an inhibitory signal is coming into play. This blocks the CD28B7 interaction, and eventually your entire signaling process is going to be stopped by the second or third day as the body wanted. This prevents your unwantedly prolonged immune response of your T cells. Now, again, an experimental way of by how people have understood this. So, since we saw that the TCR engagement causes the induction of CTLA4, right? So, this is not something very important, but just try and understand this. Okay, also, if in presence of signal 1, so let's say this TCR got an engagement signal with the processed antigen and MHC molecule, but this signal is absent as we can expect by the second or third day of T cell activation. This is absent. So in presence of signal 1 and absence of signal 2, we have the T cells getting into a phase of clonal energy. Again, clonal energy, we have also discussed this in your B cells. So energy in general, it means that there is a state of non-responsiveness. So what happens that if you, so MHC expression is already there, but your B7 expression is not here. It has been purposefully limited by fixing the cell. So fixing the cell is going to kill the cell while maintaining its structural and characteristics intact, right? So because of this interaction, we already had the MHC class 2 molecules present, but because the cells have been fixed, there is no expression of B7 that eventually takes place. As I was saying, your macrophages, they have very low expression of class 2 MHC molecule, and B7 is altogether not there. So if I take off macrophage, I fix the cell, and I now allow it to interact, because before fixing, antigen was already there, antigen processing has happened, and the antigenic peptides are also presented along with the class 2 MHC molecule, and this cell is now waiting for an appropriate T cell to come recognize this antigenic complex, and then it would have increased the expression of B7 on its surface. But since we have fixed the cell, the B7 increase could not happen. And because of that, we have no second signal possible here, because no B7. This signals the T cell to produce energic genes. Energic genes, that means that eventually the T cell is going to not give any response, right? So one way of by which energetic gene expressions can come in is that there is no second signal. The other possible way of preventing this second signal from happening here is that despite of B7 being present here, we can neutralize this molecule by having it bound by some other molecule. If I just say, if I like cover this over here like this, then also the interaction is not going to take place. So if I have a normal antigen presenting cells, which by the engagement of signal 1 has now started to express B7. But the CD28 molecule that has to interact with this B7 has been blocked by binding of a FAB fragment of anti-CD8 molecule. So just understand that something is now blocking the CD28 molecule, and same thing follows, that no second signal is present and the T cell gets into energy, right? So when energy, so in both the cases we have energetic genes expressed, and if either of these T cells, which is now called energetic T cells, is now getting a chance to interact very nicely, properly, where with the B7, with CD28 interaction. So signal 1 is also here, signal 2 is also here, but since the T cell is already in a state of energy because of prior failure of the signal 2, this is not going to give any response once again, right? And so this is a possible way where people try to do some negative experiment. So negatively, so there are two approaches of doing experiment. One, you do something that is going to stop occurrence of an event, and the subsequent follow-up experiment is that you now try and restore something to eventually get your prior positive response that you are getting. तो पहले कुछ हो रहा था फिनोमिना, इसमें आपने कुछ ऐड किया जिसकी वजह से अब फिनोमिना नहीं हो रहा. Now you try and restore something and then see that if the phenomena is again happening how it was already happening in the previous case. तो अब लोगों ने क्या किया कि ठीक है, एनर्जिक अगर टी सेल हो गया तो देयर इज नो रिस्पॉन्स कमिंग अप. Now they have chosen a fixed antigen presenting cells like over here, but along with that they have also introduced a non-fixed antigen presenting cells from the same host, that's why it's allogenic. So allogenic is something that is from the genetic makeup of it is exactly the same as is the case over here, to understand it in easy words right now. Okay, we have one fixed APC, but we have certain other allogenic APC which can give this co-stimulation. सो सिग्नल वन आ गया, एंटीजन एंटीबॉडी इंटरेक्शन मुझे यहाँ से मिल गया, सॉरी एंटीजन एंड टीसीआर का इंटरेक्शन आई गॉट फ्रॉम हियर, and co-stimulation I can get from some other allogenic antigen presenting cell. Eventually I have the signal 1, I also have the signal 2. This is going to increase the transcription of IL-2 gene, and eventually the cell is going to produce IL-2. We were talking about IL-2, that IL-2 is an important cytokine that is getting a chance to bind to its receptors present on the same cell, and this binding stimulates the proliferation of the helper T cells, right? So in this case, because both the signals are there, although they are being produced by different cells now, but this T cell only wants कि मुझे दोनों signal मिलना चाहिए, right? And eventually we have the third signal also coming into picture. Now, so instead of allogenic antigen presenting cells, we could also have the CD28 molecule co-stimulation happening by any other molecule that can cross-link these two individuals. So the idea is that this has to be cross-linked by something. If it is B7, that's okay. But if not B7, if there is certain, say, antibody designed against the CD28 molecule, if this also comes and bind, this T cell is going to have sufficient signal 1 and signal 2 to eventually produce signal 3, right? Okay. So I hope until here we are all clear.
Yes, ma'am.
Okay. So we will come to that whatever is the mechanism of T cell transduction, the details of it in a while. I just kept it here, but it's just diagrammatic go through. Let's just understand something that requires more attention, and then we will come back to this, right? So we understand that there is T cell activation has occurred. And we also have been talking about that there are two T cells, the double positive cells differentiating into T helper cells or the CTLs. Now let's just try and understand that how these two cells, once we have a CD4 cell or a CD8 cell, they can also undergo further differentiation, which is particularly of importance in your T helper cells, because if you remember, even your T cells show secondary immune response just like your B cells, right? So we had certain memory B cells forming there and some effector B cells. Effector B cells were your plasma cells, and memory B cells said whenever the same antigen is encountered again, we have a faster response. Similarly, for your T cell, we have a faster secondary immune response, which means that every T cell, apart from its effector function, is also forming a memory T cell. So we'll try and understand how that happens. And prior to this, we also have the cells differentiating into different effector subtypes, right? Which means that we have a T helper cell that forms a effector T cell. So effector T cells can be of all these different types, and we also have a memory T cell, which can again be of all these different types, right? Okay, so with the naive T cell, this recognizes an antigen MHC complex, which can be on a target cell if you're talking in context of TC cells, target cell, because the TC cells are going to recognize a cell for killing, or on an antigen presenting cells, which are so which cells recognize an antigen MHC of APC is your T helper cell. So whenever this recognition is happening, a naive T cell is going to enlarge into a blast cells and eventually re-enter the cell cycle and undergo clonal expansion. So clonal expansion that is happening, it is happening after your thymic selection has occurred. Thymic selection produces mature naive T cells, and in presence of an antigen, now this cell, like the B cells, also have to undergo clonal expansion. So for clonal expansion, what is important, that the cell cycle re-entry has to happen. So like we saw centroblasts, and from there we had centrocytes coming up. We have similarly blast T cells from which are going to enter the cell cycle once again and eventually develop into effector T cells or memory T cells for both your TH as well as your TC lineages. So they enter the cell cycle, there is induced expression of IL-2 gene, because we just saw that there is a positive feedback loop. The same cell is producing IL-2, and during its course of development, it has assembled the IL-2 receptor already. So, along with the cell cycle re-entry, it is also important that the IL-2 gene expression levels has to go up, and the alpha chain that had to form during the process of development, the alpha chain of a higher affinity now replaces the initial alpha chain that was associated with the beta chain of IL-2 receptor during the time of development. So this is how somewhat the affinity for not the antigen but the responsiveness of the T cell is better, or the clonal proliferation part is taken care of for your T cells. So this is going to signal the cells for proliferation and differentiation, and eventually effector T cells and memory T cells can form. Now the T helper cells, coming to T helper cells only, they can differentiate into 5 subpopulation. TH1, TH2, these two remains the major highlight of discussion since a long time. So most of the information that we have, so initially the classifications were only in TH1 and TH2 cell type. Eventually people discovered that we also have other T helper cell subtypes, which is your TH17, follicular T cells and regulatory T cells, right? Now, if you look here, your TH1 and TH17, these two subtypes enhances cell-mediated immunity and inflammation, which means the function of CTLs as well as the functions of your innate cells. T2, TH2 and follicular T cells, they enhance your humoral immunity complex. And as the name suggests, the regulatory T cells, they inhibit your T cells, right? So inhibit which T cells? The T helper cells as well as the cytotoxic T cells. So all of these cells are all CD4 positive and eventually have different other surface molecules, right? This T-Rex cells, they express CD25 molecule on its surface. So maybe someone might ask you that what is a specific marker for T-Rex, because these are extensive areas for ongoing research in immunology. So CD25 is a marker that is used to isolate and identify your regulatory T cells. Oh, we are already past time. Let's just try and finish this, and maybe then the CTLs we can get into it the next class, right? Okay. So if you now look here, the cytokines that are produced by T helper cells, TH1 and TH2 are different, and different cytokines obviously mediate different effects, right? So TH1 cytokine, they are primarily responsible for a way of immune disorder, which is hypersensitivity. So for delayed type hypersensitivity, it's the TH1 derived cytokines that play a role. And these cytokines also affect your CTLs, the NK cells, and the macrophages. So, which all cytokines are produced by TH1 cells? The IL-2, IL-3, interferon gamma, and TNF beta, right? So, IL-2, it helps in T cell growth. IL-3 helps in hematopoiesis. Interferon gamma activates the macrophages, and TNF beta is involved in inflammatory response, which means the cells of your innate immune system. Okay. Now over here, so these are just some things for you to remember. Now, TH2 derived cytokines primarily have the role in B cell activation. So again, IL-3 produced here is, so IL-3, if you see here, is produced in from both the cell types, right? And both of them are involved in hematopoiesis, because ultimately whichever is the cell coming up, they are all coming by a reinduction of hematopoiesis, right? So the cell number in the bone marrow has to increase. So IL-3 is involved in hematopoiesis. IL-4 and 5, they both activate B cells. IL-10 inhibits the macrophages, and IL-13 is a B cell growth factor. So again, it's going to help in the differentiation of your B cells. Now, there is one important phenomenon happening over here, which is the phenomena of cross-regulation. what happens that of all these cytokines, we have one cytokine released by either subtypes. So both these subtypes wants to maximize its number while trying to eliminate the other subtype, right? So this process where one is the cytokine from one is negatively impacting the cytokine from the other, this phenomena is known as cross regulation. So, which cytokine from Th1? It's the interferon gamma that inhibits Th2 proliferation. And similarly, we have IL-4 released from Th2 that downregulates the secretion of interleukin 12 by the antigen presenting cells. So if interleukin 12 by antigen presenting cells goes down, the Th1 differentiation is also going down. So IL-4 produced here, which functions to activate B cells, but also downregulates the IL-12 from the antigen presenting cells, and this eventually halts the Th1 differentiation. Similarly, the interferon gamma. So this way, there is a positive feedback mechanism. So interferon gamma is going to signal more cell differentiation into Th1 type, whereas IL-4 is going to signal more cells of Th2 types, right? So there is a positive feedback mechanism, and as well as there is inhibition of production of the other subtype. So this process overall is known as cross regulation. Anything from here? So this we discussed for the T helper cells. Now similarly, for your TC cells, we have them differentiating into the cytotoxic T lymphocytes. So CD8 positive cell, the TC cells, they further differentiate into CTLs, which can be of an effector type or of memory type. So as the name suggests, the CTLs, we have been talking about it, that they kill the target cell. And which all can be the target cell? It can be a virus or bacteria infected cell. And so these are the endogenous pathogens. They get their peptides processed in the endogenous through the endogenous pathway. And eventually the MHC1 molecule displayed on the surface of these target cells are recognized by the TC cells. They are then going to differentiate into CTLs and kill about the cell, or it could be certain altered cell or tumor cells that are also the target cells for your CTLs. CTLs also synthesize certain cytokines that can augment these functions. So if it is, we can have a CTL synthesizing interferon gamma. This is going to further facilitate the killing of virus or bacteria infected cells, and TNF beta primarily has a role in further killing of your tumor cells. Okay, now let's just come to this that eventually it's now let's just try and generalize things back together. We saw for T helper cells and TC cells in separate context, but the process of differentiation of these we discussed the effector cells for both of these lineages separately, but a matter of fact remains that the effector cells are short-lived, which means from few days to few weeks, and that's it. But the progeny cells that differentiate into memory cells can be long-lived. So for both the cell types, we can have some memory cells which are long-lived compared to the effector T cells. Right? Okay. So another fact is that we tried to discuss it over here, that some cells have constitutive expression where some have induced expressions, and so different antigen presenting cells, what we could understand is they have a different role in stimulating your T helper cells, right? Because all antigen presenting cells are eventually giving a signal to your T helper cells, provided if them on their own they are not having some viral particles inside of it, right? In that case, they can also stimulate your cytotoxic T lymphocytes. Because all antigen presenting cells on itself is also a nucleated cell, and every nucleated cell can express a class 1 MHC molecule. Now, if you look here for dendritic cells, the dendritic cells have constitutive expression of class 2 MHC molecule and constitutive expression of B7 molecule. So thus they can activate your naive T cells, effector T cells, as well as your memory T cells, right? Because so what is not there in your naive T cell? The naive T cells and in the first go do not have the CD28 molecule that is present, and it gets to express only in so the number of CD28 molecules is increased in presence of an antigen in complex with your MHC molecule. So naive T cells, as it expresses now the CD28 molecule, we already have the B7 present here. So the interaction, the co-stimulation is faster. So the naive T cells can be activated by your dendritic cells. Now, if you look for macrophages, a resting macrophage, so they can and the uptake of antigen by dendritic cells can happen by endocytosis as well as phagocytosis. But for macrophages, the uptake of antigen happens in a phagocytosis manner. Now there are two possible stages of macrophages, the resting state as well as the activated state. So resting state is where we have no antigen encounter by the macrophages and no expression of antigen on its surface. So in that state, there is no MHC2 molecule, no B7 molecule, and hence it cannot activate any of the T cell subtypes, right? But the expressions are inducible. So as soon as a antigen gets inside the cell, say by phagocytosis, it is processed and along with the MHC2 molecule, the levels of it somewhat increases, but it is still lesser than the levels of MHC2 on dendritic cells. We have induced expression of MHC2 molecules that present the peptides along with induced expression of B7 molecules. So now they can so initially when the encounter of a T cell happens, so this T cell is going to be a effector or a memory T cell, because naive T cell activation is only possibly happening for dendritic cell, because here it cannot get a chance and interact because we do not have anything present on it. But your effector T cells, which are already formed by the time your MHC2 and B7 expression is happening on your macrophages, these are the primary group of T cells that are getting activated by your activated macrophages. Similarly, for your B lymphocytes, we have constitutive expression of MHC2, but again lesser than what is compared to dendritic cells, and there is an inducible expression of B7. So initially there is no B7. So here we have the effector T cells and the memory T cells that can still somewhat get activated because of their interaction possible with the MHC2 molecules, right? But because they already recognize the antigen because of its prior memory of it, the prior response of it, they are able to recognize this antigen. So effector T cells and memory T cells can be activated by your resting B lymphocytes. Now, by receptor mediated endocytosis, if it has taken up an antigen, there is now increased expression of MHC2 comparable to the level of MHC expression on your dendritic cells. Alongside that, we have the B7 expression, which has also increased. So since now B7 as well as MHC2 molecules are present here, we can have the activation of naive, effector as well as the memory T cells happening.
Excuse me, ma'am.
Hanji.
Antigen presenting cells are having both the classes of MHC molecule present on them.
Every cell has MHC1 molecule. It is only the MHC2 molecules present only on the antigen presenting cells.
Okay, ma'am. So, ma'am, in the resting stage also of macrophages, it is having MHC class 2 molecule present.
See, in the resting class, it is not present as it is also mentioned over here. So the expression is not constitutive, but the expression is inducible. So the other cells of your body, let's just say maybe your WBCs, some other WBCs, maybe the T cells in itself, they do not have a possibility also where a MHC2 molecule can get expressed. But in present of an internalized antigen, there is a possibility of MHC2 expression that happens. So the expression is inducible. But that doesn't change the fact that they are antigen presenting cells, because if activated, they can present the antigens. Even at some point of time, they have the possibility of MHC2 expression, they have been now classified as antigen presenting cells.
Okay, ma'am. And ma'am, B7 is presented only by APC cells and not by all the cells of the body. Am I right?
Yes, B7 is also a possible marker for your antigen presenting cells.
Okay, ma'am.
Okay. Okay. So So along with all these interactions, now if again from the previous class, if you so the binding of antigen and antibody is a very strong interaction, but we discussed that the binding of your TCR to your antigen MHC complex is comparatively of a lesser affinity. So just when the TCR is getting a chance of binding to the antigen MHC complex, this T cell also has a upregulated expression of cell adhesion molecules, just to ensure that this TCR antigen MHC binding is strengthened, which means कि आपका जो एक सेल है इसके पास एक कुछ है बाइंड करने के लिए, but now it before it like mediates a response, it do not want it to lose the eventual interaction with the other cell. So what it does, it produces certain adhesion molecule. It upregulates some adhesion molecule. So some of the adhesion molecules are upregulated on your antigen presenting cells, while some of the molecules are upregulated on your T helper cells to strengthen this interaction of TCR antigen MHC further. Just certain names over here. So CD54, which is also called as ICAM1, and CD58, also called as LFA3. These are expressed on your APCs. They respectively interact with integrin or CD11A or CD18. So just some theoretical factual details over here, which are present on your T helper cells, and this interaction further stabilizes the interaction of your APCs or the target cells with your interacting T cells. Right? Now, if this is so far clear, and if we do not have further question, we can quickly go over this diagram. Or if we have some questions, we'll take that first. Okay, let me just go through this. And this is the signaling part that we have discussed in context of B cells, and we also discussed this quite some detail the last class. So what happens? It's a very clear, neat and clean diagram that there is an engagement of MHC peptide, right? So this is class 2 MHC peptide being presented by your APC, and this is a T helper cell. So if you look here, class 2 MHC along with the peptide is getting presented to the TCR. Along with the TCR, we have the associated CD3 molecules, right? And we have a co-receptor here, the CD4 co-receptor, or it could be the CD8 co-receptor based on, but they have mentioned it to be class 2 MHC, so this essentially has to be a CD4 receptor. Anyway, right? What is important here is the cytoplasmic tail has the docking site for a molecule called LCK. This LCK bind, binds to the cytoplasmic tail of your co-receptor molecule, and this phosphorylates the ITAM. So ITAMs we were discussing in the last class also. This phosphorylates the ITAMs of your signaling molecules associated with the TCR. And if this phosphorylation is happening, so we have okay, look here. So this phosphorylation is additionally creating a docking site for some other molecule called as ZAP70. I don't think this signaling is a part of your regular course, but just for your understanding. Now, if the ZAP 7 tails binding, some factors is changing to certain other factors, and eventually we have certain effector molecules produced. PIP, phospholipase C, GEF, all of these are signaling molecules that are very, very nicely discussed in the courses related to cell biology, cell signaling mechanisms, right? So, what do they bring about? So, if we have any of these pathways activated, they are going to further produce secondary messengers. These secondary messengers can either can mediate different pathways, right? So it can be a PKC mediated pathway if the activation is through PIP and IP3 pathway. If it is a DAG pathway that follows from PIP, it could be certain intracytoplasmic calcium changes that happen, and because of the calcium, certain other changes in gene expression levels or certain killing functions that we will discuss in the next class, those things can be mediated. If it is a GEF mediated pathway, some small G proteins can be activated, and RAS and RAC pathways are downstream of your G protein coupled receptor. Eventually, by either of these pathways, the goal of changed gene expression, certain functional changes, differentiation, and activation follows in your T cells or T helper cells or your cytotoxic T cells. Right, so just a signaling mechanism. We will not get into the details of how exactly all the mediators convey the signal, because those are relevant to certain other courses. But we just have to understand that whatever is happening as a matter of fact here, it is translated in the inside of the cell, and eventually the cell, if we say proliferation is happening or differentiation is happening or something is upregulated or something is downregulated, it is happening by certain intracytoplasmic signaling mechanisms. Okay, so we'll stop our class for today over here. Shamika, if there's anything that we have to ask, then we can go over it. And we will keep the discussion related to CTL, related effector responses, for the next class.
Ma'am, can you just show me the mouse experiment that you have demonstrated me?
Over here.
Ma'am, here in this case, I have asked you some doubt. I'm not remembering that. So, one of that reason is can might be possible for that question answer is that it is not having the MHC molecule being presented by some other cells to it. So, in that case, too, it is not able to kill that strain A cell.
Which case are you talking about? Are you talking about here?
I have asked you that it is not
Mm-hmm.
having CD4 positive cells, right?
Mm-hmm. हाँ, you were asking that if this is for CTL, so CD4 positive cells नहीं है यहाँ पे something. Mm-hmm. Go ahead.
So, ma'am, one of the reason for not being presence of there is might be that it is मैम क्योंकि वहां पर एंटीजन मैम एक्चुअली मैं क्वेश्चन फ्रेम नहीं कर पा रही हूँ इट इज
Mm-hmm. सी अगर आप
मैम
आर यू ट्राइंग टू से कि यहाँ पे किलिंग नहीं हुआ, उसका एक रीजन हो सकता है कि एमएचसी रेस्ट्रिक्शन है, और यहाँ स्ट्रेन ए के साथ अगर हमने रिएक्शन होने दिया तो स्ट्रेन ए पे एमएचसी ऑफ स्ट्रेन ए टाइप की है, but the education of the CTLs have been for strain B. That's why they could not react over here and there was no killing. Now I think you are trying to ask that possibly CD4 positive cells भी यहाँ पे नहीं थे इसलिए किलिंग नहीं हुआ? इज इट समथिंग लाइक दैट?
मैम क्योंकि CD4 पॉजिटिव सेल्स के लिए MHC मॉलिक्यूल्स को एंटीजन प्रेजेंट करके दिखाना, दैट मस्ट बी द नेसेसरी कंडीशन, बट वो यहाँ पर नहीं है। सो इसीलिए मैं सोच रही थी कि यहाँ पर CD8 पॉजिटिव सेल्स ही जो है, दैट्स व्हाई दे आर एबलिंग टू किल दैट स्ट्रेन बी सेल्स।
सी अगर आप चलो ऐसा एज्यूम करते हैं कि यहाँ जो स्प्लीन सेल्स है वहां उन्होंने सिर्फ सीटीएल्स की बात नहीं करी, सीटीएल्स को भी CD4 पॉजिटिव सेल्स से सिग्नलिंग चाहिए होता है, राइट? बिकॉज दे आल्सो नीड साइटोकाइंस टू ब्रिंग अबाउट द किलिंग फंक्शन। नाउ इफ यू आर सेइंग कि CD4 पॉजिटिव सेल्स भी थे यहाँ पे, और यहाँ पे CD4 पॉजिटिव सेल्स एपीसीज के साथ इंटरैक्ट नहीं कर पाए इसलिए किलिंग नहीं हुआ, है ना? तो यहाँ पे CD4 पॉजिटिव सेल्स एपीसी के साथ इंटरेक्ट क्यों नहीं कर पाए? इसीलिए नहीं कर पाए फिर से क्योंकि एमएचसी यहाँ पे ऑफ द एंटीजन प्रेजेंटिंग सेल्स विल आल्सो बी ऑफ योर टाइप स्ट्रेन
स्ट्रेन बी।
ए।
मैम स्ट्रेन बी का होगा ना, क्योंकि वहाँ पर थाइमस जो है वो ग्राफ्टेड जो हुआ है वो स्ट्रेन बी से हुआ है।
इसमें बोल रही हूँ मैं ऑफ दिस केस। यहाँ पे अगर हमने स्ट्रेन ए के सेल्स लिए, मानो अगर हमने स्ट्रेन ए के एंटीजन प्रेजेंटिंग सेल्स लिए, तो स्ट्रेन ए के एंटीजन प्रेजेंटिंग सेल्स के ऊपर क्लास 2 MHC मॉलिक्यूल जो होंगे वो भी स्ट्रेन ए से आए हुए MHC मॉलिक्यूल्स होंगे, राइट? बट यहाँ के जो CD4 पॉजिटिव सेल्स है वो स्ट्रेन बी से एजुकेटेड है, स्ट्रेन बी के क्लास 2 MHC के सेल्स है, इसलिए भी वहां पे इनका इंटरेक्शन नहीं हो पा रहा है।
यस मैम। सो मैम वो रीजन ये भी हो सकता है ना उस क्वेश्चन का।
See, yes, but then the killing function that they have talked about specifically. So two things here. इन्फेक्टेड माइस में से हमने स्प्लीन निकाला, तो यहाँ पे जो स्प्लीन के सेल्स है वो ऑलरेडी effector T cells होंगे। And what is the effector response that they are trying to discuss is killing response. So killing by effector T cells present in the spleen of your previously infected mice, right? तो ये अगर इफेक्टर है तो इनको ऑलरेडी पता है, बिकॉज दे हैव दिस एलसीएम वायरस इन्फेक्शन की, उनके उनको नॉलेज है, दे आर ऑलरेडी एजुकेटेड अबाउट दिस। एंड नाउ व्हेन दे आर अगेन को इनक्यूबेटेड in vitro setup, irrespective of the presence of your CD4 positive anything, because okay, culture medium they might have put something, but if they are talking about killing is something that they are quantifying the mechanism of your CTL। इट इज किलिंग को अगर हम लोग एक्सपेरिमेंटली एवेल्युएट कर रहे हैं तो हम CTLs की ही बात कर रहे हैं, नॉट वी आर नॉट टॉकिंग अबाउट CD4 बस।
ओके मैम, आई एम नॉट हैविंग एनी फर्दर डाउट्स।
Okay, so let's just meet each other the next class with more fresher energy. We will complete our discussion of T cells in the next class. We will also try and complete cytokines, and then we will quickly discuss the immunological techniques, right? So immunological techniques में आप लोगों को recorded classes में I think facts और immunoprecipitation, ELISA ये चीजें बताई गई है। Those are comparatively straightforward topics, so we will go through very quickly. So cytokines is again a very theoretical topic, nothing much to understand there. Our understanding related classes were primarily for your B cell, T cell, and then we will have one class where we discuss all the antibody effector mechanisms. So these three topics are the most understanding intensive classes. Apart from that, cytokines and immunological techniques we will do in the next class, which is very information centric classes. Nothing much, nothing very heavy. But yes, we will hopefully complete until there by the next class. Okay, so I can end the session.
यस मैम। मैम
Uh,
आई वांट टू आस्क यू दैट which book can I refer for cell biology?
for cell biology, I am not recalling the name right now. I'll get back to you in the next class. right? I think Robert, some book named as Robertson. I think that is the book that is the best reference for cell biology, but I'll get back to you in the next class. You can also refer to Watson. So molecular biology के लिए Watson recommended होती है, बट molecular Watson की जो बुक है उसमें सेल बायोलॉजी के भी काफी concepts शायद discussed है, if I'm not wrong. So I'll get back to you.
Okay, thank you so much, ma'am.
Okay, good night.